All right, so this video is to catch up in section 3. So if you missed these pictures, I can draw them for you and you can get them into your notes. So our objectives here are to talk about light-dependent reactions and the light-independent reactions. So we're going to talk about light-dependent, which is going to be my electron transport picture. And then we're going to draw another picture with the light-independent reactions where I have my Calvin cycle. So I need to connect light dependent with electron transport and light independent with Calvin cycle. But I have two main big pictures to draw for this that if you understood those pictures, you could understand this section pretty well. So our first picture, we're going to draw that electron transport. So I just start out by drawing phospholipids in a membrane, because that's what this is. Again, is the membrane of a thylakoid that I'm talking about here. And then I'm going to have this protein here that is green, right? This is my chloroplast. That's going to absorb some sunlight energy. So I need a little wave coming in here, some light energy coming in there. That's going to be absorbed by electrons inside this chlorophyll. And then those electrons, once charged, will make their way down a transport chain. So I just have this other chain of proteins within the membrane. So I continue to draw my membrane. And like I said, electrons get charged up, they jump out of the chlorophyll, and they make their way down this path, right? Now, that may seem kind of weird at first, but essentially, I'm just creating a current. I have electrons that are flowing continuously, jumping out of that chlorophyll and moving down this electron transport chain. In this membrane here, and I'm not going to draw it because it takes up a lot of space, I'm going to have something that I'm going to power these pumps that would push hydrogen ion across the membrane, right? So if I had some hydrogen ion down here, I can take that hydrogen ion and move it through the membrane to this side so that the hydrogen ion concentration on that side keeps getting bigger and bigger and bigger. So we'll talk about what we're doing that for, why we're moving that across. But this is definitely an active transport. I'm pushing that hydrogen ion across the membrane, and that's why I need those electrons to flow down that protein chain. So that current powers those pumps that push hydrogen ion across the membrane. Now, I can't continue to do this forever because chlorophyll runs out of electrons. But luckily, I have a molecule in my body, or there's a molecule, in a plant's. I keep getting the wrong color there. There's a molecule that a plant can break down, water, that will give me those two hydrogen ions, right? Some of which I'll push across the membrane, and half an oxygen molecule, right? Water needs to be split in a plant in these chloroplasts, in these thylakoids, essentially just to replace the electrons that are lost by chlorophyll. So again, this is only going to work, this whole process, pushing hydrogen ion across the membrane is only going to work as long as I have electrons to move down that transport chain. As long as I have electrons in this chlorophyll to absorb light energy and start moving down a chain of proteins, I can do what I need to do. But chlorophyll doesn't have an infinite amount of electrons. They have to be replaced. So splitting the water is really important because it replaces those electrons lost. So this is essentially all of photosystem 2. So you need a Roman numeral 2 in there. Photosystem 2 is followed by photosystem 1. Not really sure why they went with that order, but you can't really argue with me about it. I didn't make that decision. But I have another chlorophyll molecule here at the end of that first chain of proteins. So my electrons from photosystem 2 end up in photosystem 1 over here. Obviously, I got green again for a reason because I need to absorb more light energy. So I got another wave coming in there. And that light energy is going to continue those electrons along a similar path. There's another chain of proteins over here. Again, just kind of fill in my phospholipids to continue making this look like a membrane. Your picture doesn't have to be perfect because obviously mine isn't, but at the end of this transport chain, so the electrons absorb new energy, can travel down this chain, but at the bottom of this chain, instead of powering pumps, I actually just catch those electrons in this new molecule, this NADPH molecule, that I can make from a NADP plus molecule, right? If I take this NADP plus and a hydrogen ion, I can catch 2 electrons in this new energy molecule, NADPH. So really at the beginning here, just for accounting purposes, there are 2 electrons that come out of that water. Those 2 electrons move along this whole thing, 1 electron at a time, really. But ultimately, I catch 2 electrons in that NADPH, and that NADPH is going to be an energy molecule that I can use later, right? Kind of like ATP. The energy in that NADPH more or less comes from the light energy here from photosystem 1, almost directly. Otherwise, I will capture a lot more energy in this process, but it's kind of happening somewhere else, right? So I need to add another protein to my membrane, and I'm going to put it over here just for a lack of a better option. So you can put it on the right or the left, it doesn't really matter. All the rest of the picture had to be connected together because photosystem 2 and 1 are literally connected to each other. This channel protein really could be anywhere in that thylakoid though. So I'm going to take advantage now of this hydrogen ion concentration that I've built up. The next step of the process, the last step, is I need to let this hydrogen ion flow right back through the membrane, but only in specific places, right? I have this protein here that's actually going to be labeled. Let me put a label on this. So just use like blue arbitrarily, right? This protein is going to be called ATP synthase. And it's a special transport protein that not only allows that hydrogen ion to go through, but can turn essentially like a turbine, and it can create energy from that flowing movement of hydrogen ion. It can take that energy and turn it into a different form so that I can create a bunch of ATP over here. So it's basically just a machine that makes ATP, but it requires hydrogen ion to flow through it to make that happen. So this next part here is a little bit more convoluted. You got to recognize that this light energy here is what powered that active transport across the membrane so that I could build this huge concentration of hydrogen ions so that it can come back through this ATP synthase and I can make a bunch of ATP. So that's not quite as direct as the light energy in photosystem 1 ending up in NADPH, but light energy in photosystem 2 ultimately is used to make ATP. All right, so that's a good enough picture for our electron transport in these notes. So I'm just going to quit there and make another video for the other picture that we need.